Good morning. I'd like to thank everyone for being here. So as George mentioned, I will be sharing with you my summer project here that dealt with analyzing cold responses in Solanum, lycopersicoides, and introgression lines. And I was interested in finding those introgression lines that could potentially be cold tolerant. So I'll start off by giving you some background into Solanum, lycopersicoides, as well as its introgression lines. Then I'll mention some of the experimental methods that were used throughout my project and the results that were collected from them. I will also go into the future work that would be interesting to see done later. So the tomato industry is made up of both the fresh and processing tomato industries, estimated at about $2.6 billion annually by the USDA. And already favorable traits have been developed through continued breeding, and these would include things like delayed ripening, which allows for easier transportation of crops, as well as pest resistance, which essentially increases a field's productivity. Other phenotypes are still in the research and developmental stages, such as cold and drought-tolerant crops, and these are of interest because they would essentially make crops more resilient. So Solanum lycopersicoides is a nightshade tomato relative native to Chile and Peru. It is actually the most distant tomato relative able to produce a viable offspring when it is hybridized. And it is also the nightshade that grows at the highest elevation. And recently, its genome has been sequenced here at BTI. So here's a little bit more information into the environment which Solanum lycopersicoides grows. So here we see in this graph Solanum lycopersicoides being subjected to below freezing temperatures on average, compared to the ancestor of the cultivated tomato here, shown in orange. It is subjected to above 15 degrees Celsius. So we can see a big change between Solanum lycopersicoides and the cultivated tomato. Furthermore, it grows in a rather dry environment here in Chile and Peru, in the area of Chile and Peru. So collectively, this makes Solanum lycopersicoides and its introgression lines of interest to both cold and drought-tolerant studies. So Solanum lycopersicoides introgression lines have been developed by researchers at UC Davis through consecutive backcrossing. So what happens is that they take Solanum lycopersicoides, cross it with the cultivated tomato to produce F1 hybrids. These F1 hybrids are then back-crossed to the cultivated tomato, and that leads to the development of introgression lines. Each of these introgression lines has unique chromosomal segments coming from the parental lycopersicoides into the genetic background of the cultivated tomato. And each of these chromosomal segments may be conferring some type of phenotype that would be of interest and may only be shown in one or a few of these introgression lines. So in my study, I used a homozygous subset of an IL population to be able to determine the genes that may be conferring these tolerances with ease. So studies like these are of relevance to agriculture, given the growing world population and the changing weather patterns due to climate change. There's an increasing strain on agriculture. So these studies have the potential of introducing quantitative trait loci, or QTLs, that are maybe conferring cold tolerance into the cultivated tomato. And again, as I mentioned before, cold and drought-tolerant crops are more resilient, so they would be better able to withstand harsher conditions. And they would also be able to extend the seed-to-harvest window, which would essentially means being able to seed at a later date and still have enough time to harvest. So that would not only allow farmers more flexibility, it would also increase productivity as well as profitability. Now, here are the steps to going about doing that. So as we see here, the first step would be analyzing a population after cold treatment, identifying those lines of interest, finding the potential cold-tolerant QTLs, introducing them into the cultivated tomato to lead to cold-tolerant crops. And in my study here, I focused on these first two steps. So this is a paper that was published in the Science Journal, which showed a calmodulin-like gene conferring tolerance to Solanum habrocaides, which is another wild nightshade relative. So here we see that the overexpression of this calmodulin-like gene was conferring cold tolerance. As we see here, after treatment, the wild type was greatly affected, whereas the lines that were overexpressing this gene seemed to be more tolerant. So this qualitative data was further supported with quantitative data. They tested levels of MDA, proline, chlorophyll, and electrolyte leakage. So this summer, I decided to do something similar. So melanodialdehyde is essentially a product of lipid peroxidation. What that means is as reactive oxygen species, or ROS, accumulate in the lipid bilayer, they start degrading lipids there. And what ends up happening is that that reaction leads to the formation of MDA. Proline is an amino acid that has also been shown to be an osmolyte, a chaperone, and a signaling molecule. Chlorophyll has been well documented to be degraded under oxidative stresses such as cold stress. And Brix, which is essentially a measurement of the total soluble sugars, basically has been shown to be a documented fruit stress response in the Giovannoni lab. I also conducted a survival assay, which would further support all of our quantitative data here. So my goals for this summer were to modify and optimize protocols to determine stress in this IL population. I also wanted to identify those lines that could potentially be cold tolerant. And my hypothesis was that cold tolerant ILs would exhibit lower levels of both MDA and proline, as well as showing a more stable chlorophyll content and displaying less signs of wilting, chlorosis, and necrosis in the survival assays. So 2 rounds were conducted throughout my experiment. Round number 1 was first ran on the cultivated tomato, and round 1 consisted of a 12-hour, 4 degrees Celsius treatment on 5-week-old seedlings with a 2-day recovery period. And we saw a significant decrease in chlorophyll, as we can see here. And that supports the presence of oxidative stress. If we look at the qualitative data here, we see that after cold treatment, it seemed to have minor signs of wilting, but overall not very much difference from the control. Now, we did not see any significant changes in neither MDA, proline, or Brix. When we look at the whole population, we see an increasing trend in MDA, which is as we would expect given the oxidative stress. Now, those introgression lines that are boxed in orange show lines that are either having not a significant change at all or decreasing in MDA, which we would expect if those for lines that are cold tolerant. And the lines with arrows pointing at them will be lines of interest throughout the assays, so we'll follow these through. When we look at proline, it's a much more scattered trend, but again, I have boxed those lines of interest. These are lines that are either showing no significant change in proline or are showing a significant decrease in proline, which we would expect if the lines were cold tolerant. And again, here are our 3 lines of interest. When we look at chlorophyll, we see an overall decreasing trend in chlorophyll. Again, I have boxed those lines of interest here. Now, please note that since this is chlorophyll, we expect it to go up versus going down. We expect it to not be degraded. And again, we have our 3 lines of interest here. So these would be lines that are either increasing in chlorophyll or having no significant change in chlorophyll. Now, we decided to run round number 2. We did this to be able to see if we could stress the cultivated tomato out a little bit more. So essentially, round number 2 consisted of a 7-day, 4 degrees Celsius treatment on 3-week-old seedlings with no recovery period. So I figured that this would not only increase oxidative stress, but also have the highest levels of these molecules directly after that treatment. So here we saw a significant change in both proline and chlorophyll. As we can see here, proline significantly increased after cold treatment, and chlorophyll significantly decreased. So this supports the conclusion that this cultivated tomato is undergoing oxidative stress and it is showing signs of cold sensitivity. When we look at the survival assay, we can see that the treatment cultivated tomato is showing signs of chlorosis as well as necrosis, and it also showed stunted growth. Now, when we looked at the whole population in the survival assay, we saw definite indications of stress. There was significant wilting, chlorosis, and necrosis in most lines. But when we pulled out our 3 lines of interest here that we had identified in the previous assays, we saw what seemed to suggest lower levels of stress. There was little to no visible wilting, chlorosis, necrosis in the survival assays. And as we can see here, 4310, 4299, and 4238 all seem to be rather similar to the time 0. Furthermore, 4310 seems to have slightly expanded its leaves, and it seems to be a little bit of a darker green. Now, 4238, although we only had 1 replicate, showed signs of a lot less of a response to the cold treatment compared to other lines. Now, in summary, the lines 4310, 4299, and 4238 could potentially harbor cold-tolerant QTLs. And again, this conclusion has been supported by the results from all assays, including MDA, proline, chlorophyll, and the survival assay. So some of the future work that could be done is conducting these similar cold stress experiment under lower temperatures. So as I mentioned before, Solanum lycopersicoides is subjected to below freezing temperatures at some point in the year. So maybe conducting this experiment under those conditions would increase oxidative stress and may give us a clearer picture. Also, finding the specific QTLs that may be conferring this cold tolerance would be of interest, and that would be done through gene expression assays. I'd like to acknowledge Dr. Jim Giovannoni for allowing me to work in his lab, my mentor, Lance Tail, as well as all the Giovannoni lab members, Dr. George Jander and Delaney for putting together this REU, and the funding that came from the NSF, the USDA, Cornell University, and the Boyce Thompson Institute. And with that, I welcome any questions. Yes. Yes. Mm-hmm. So that line took a little bit longer to germinate, so it may be that it was a slow germination compared to the others, so that might have been it, but there might be some effect coming from the cold stress that may be inhibiting its growth as well for the smaller line, 4238 you mentioned. So that could be determined by allowing it to grow a little bit longer, but in the interest of time, we couldn't allow it to do so. So we're not sure if its growth was stunted due to the cold stress. Yes?
How many of the 4238 plants do you actually grow? Maybe it was just this one plant that had growth problem.
Yeah, so unfortunately, this was the only one that actually germinated for the survival assay. The tests that were conducted or the assays that we're conducting in round number 1 actually consisted of a different population that was subjected to that 4 degrees Celsius treatment for 12 hours. So this 4238 line was only done for the survival assay. So it may be, it might have been better to see more replicates, but unfortunately, it was the only one that germinated.
So how far from marketable fruit are these introgression lines?
It's a long ways. I mean, you still have to find the QTLs and introduce them into the cultivated tomatoes. So I mean, definitely a ways away. And in the future, finding those QTLs would be of interest, and it would be something that would be of interest to me to continue.
So like the current lines are not edible?
The current introgression lines, they are, yes. So we grow them in the greenhouse, so they definitely do fruit and everything. And we have been kind of documenting their growth and their fruiting as they progress after the cold treatment. And we see some interesting trends after that.
You can bring some if you want to try that.
Yeah.